So yeah, we've got an hour actually now to talk about genomics, and if we're not careful, it could be a little bit heavy. So hopefully it'll be more of a kind of conversation. We're splitting it into two parts. The first part, Sam Rack, who some of you might have met before, who's our previous research fellow, has done a presentation, was going to present, but he's got some sort of flu-like illness. So I'm going to talk through Sam's presentation. But the first part is just to kind of talk through some ideas and concepts in genomics. The second half, Matt Church is here. So he's one of the medical oncology trainees at the Christie. And he's going to talk through specifically whole genome sequencing, which is one of the types of genomics that we've been doing at the Christie that also is sort of happening throughout the NHS. So hopefully by the end of the hour you'll have more of an idea about this sort of breadth of genomics that can be done and how it might relate to questions we've got, and what this whole genome sequencing is and how useful that might be. And one other thing, I want to say it now because otherwise I'll forget, but Matt being here is really important, right? Because what we're doing at the Christie, we've got this whole sort of practice through this group, through you lot, all the patients that come through the Christie. We've built up the kind of, the activity that we do. We have clinical fellows like Joe who are full-time, and Sam, our previous fellow, who's full-time, and we're getting more and more oncologists involved in the management and therefore understanding salivary gland cancers. So Matt's an example of someone who's kind of in the system elsewhere. So he's about to start a PhD in genomics, in cancer genomics. He's actually going to be with the experimental cancer medicines teams. This is the early phase unit who are involved in developing all new drugs. Okay, so his PhD actually isn't going to be in salivary gland cancer, but look, he's here. And for us, that's a really good thing. It's not just a good thing because he's doing a bit of work for us on the side. It's a good thing for the future. We're training clinicians in salivary cancer. So I just want to make that point at the start, because otherwise I might forget at the end. So can we put the slides up? So I'm happy if people want to interrupt at any point, just put your hands up or just kind of shout or whatever. So yeah, so this is an introduction to cancer genomics, really. I know he's put SGC, but it's just more about cancer genomics. And then I might as well tell you about Sam if you don't know, because he's put another slide up, slide 2, please. So Sam, just for those of you who have met him before, I think some of you may have met him. He's presented at this group a couple of times. He's actually been doing research projects with me since about 2018, since he was an undergraduate. He's now an academic medical oncology trainee funded by, or in part by the NIHR, so they are a kind of a core funder of future clinical academics, and he's kind of taking all of his salivary expertise into that with him, and should be all going to plan doing his PhD next year in adenoid cystic carcinoma, 3 years full time, and then just taking all of this stuff he's been doing on and changing things for the future. So that's a bit about Sam. And so, yeah, the talk, a bit of, biology revision or biology vision, if we've not done biology before, so just a bit about biology. I think it was biology revision for Sam, and then a bit about why do we need to know something about cancer genomics, and how we carry out our research into cancer genomics. Okay, so this was me talking through Sam's slides here, so a few basics. So the human body is made up of organs, like the lung, the heart, the liver. The organs are made up of cells. And I was trying to think on the tram on the way here, if I had a pin head, not the pointy bit, but the top of the pin, and if it was 1 millimetre across, which they're probably not, I was trying to work out roughly how many cells you would fit in a sort of single layer. I think you would fit about 10,000 on a head of a pin, on that, the rounded head of a pin, assuming they're about 10 microns across, which they are. So, cells are these tiny things that you can't really see, but you can see them actually if you get enough of them in a dish or in a place. And what is a cell? I mean, they sort of look a bit like that. They are made up of lots of things, including proteins. Proteins are the things that, they do things in your body. So if a cell wants to move, it has this cytoskeleton. So it's this kind of big protein architecture. It's like scaffolding inside the cell, and these things remodel and move and change the shape of the cell. So cells are made of proteins. Proteins are there because of genes. And so the kind of basic, dogma of biology is that your genes, which are a string of DNA, produce RNA, which matches to the DNA, and and it's just complementary, and that RNA encodes the proteins. And so how cells function is based on what proteins they have at any one point in time, and what proteins they have is determined by the genes. Okay, so that's kind of why genes are important. And what is a genome? So the genome is the entirety of the DNA script in your cell. So every single cell, so those 10,000 cells on the head of the pin, every single cell has the nucleus within the cell, and in that nucleus is this entire script of A's, T's, C's and G's, so this repetitive sequence that codes for that makes up the genes, which encodes the proteins, which makes the cell do what it wants to do. And I don't know if anyone's been to the Wellcome collection opposite Euston Station. I looked ages ago, I don't know if it's still there, but they used to have the human genome printed out in sort of paper form, and you could literally stand by it, and all the chromosomes were from the sort of first human sequenced, the reference genome were there on some shelves that were probably 8 foot high, 10, 12 foot across, and you could literally just pull these big thick books, open them, and it's just a load of boring A, T, C, it's all there, A, T, C, G, T, G, C, A, T, G. And there's about 3 billion letters in that code. So yeah, the purpose of this talk though is just to say we're talking about the DNA genome. Okay, so mutations. Anyone before I start to talk about, so this is now, well, what goes wrong in the DNA when we're talking about mutations? This principle of the car, so Sam's done whatever that is, a Volkswagen Polo. So mutations can kind of make the protein faster in his analogy or slower. What's his crap car? Loss of function, yeah, stop working. So you can soup up your Volkswagen Polo, or you can just kind of stop it working. Okay, so and we often use the language gain of function or loss of function. And so what that means is if you've got a protein, proteins can just basically, instead of being switched on at the right time and do their thing at the right time, they just do their thing all the time. And that's a problem, in some settings, in some contexts. So you get gain of function mutations. And so that's one thing we look for. Bits of change in this sequence that cause gain of function, cause a protein to be switched on all the time. There's loads of examples of gain of function mutations in cancer treatments. And in principle, so the gain of function promotes these things that cells do that makes them cancerous. So that's to grow, to divide, to invade the tissues, to move around the body. These are the sort of characteristics of cancer cells. Then you have loss of function mutations. So some proteins, their function is we often use the term kind of to maintain the integrity of the genome or maintain the integrity of the cell, meaning just to keep it doing what it should do. So if you lose the function, these are the caretakers in a school, and you take out the caretaker and the lights start going out and the taps start leaking and all this sort of stuff, and the caretaker's not there. So you can get loss of function mutations, that's some loss of function, then you stop taking care of the bits of the cell that you need to, and then all these same sort of characteristics start to develop, these cancer characteristics. You get variants, uncertain significance, so there's things that we just don't know what they do. And when you start looking really hard, and this might come up actually in conversations if people have ever had their genome sequenced, the harder you look, the more you find. You're just going to find more. You sequence 3 billion bits of sequence instead of just some targeted sequencing of a few genes. You start to find loads of things. And most of the things that you find when you have these, whatever you want to call it, deep dives, big thinks, hard looks, whatever you want to call it, often get labelled uncertain significance because we just don't really know. Or at least we don't know at this point in time. So why does it happen? I mean, this is one of the big questions. So there's an assumption that, and it's that the genetic change is one of the first events that causes a cell to become cancerous. But kind of something causes that, so what causes it? So one thing is you can get damage to the cell. to the DNA from external sources. So we're all kind of probably familiar with ultraviolet radiation in the sun. We all get sunburned, get a red forehead. This is because of my DNA damage in my head. Okay? And so you get external sources will damage your DNA. And normally your cells just repair. Viruses. So you can get, we don't have good examples in salivary cancers, and it was something that gets discussed now and then, is there a viral cause? In other common or garden head and neck cancers, there are, HPV virus is initiating a sort of start event for cancers. And there are others like, Epstein-Barr virus is another one in the head and neck. And then, this is the bottom one is the main thing, errors when cells divide. So actually when cells divide, it's a very, very accurate process, but when a cell divides and has to copy 3,000 million bits of sequence, you're going to get some replication errors. And so that's probably the commonest source of errors. And that's why sometimes if people say it just happens by chance, what they mean is you get these errors when cells divide. So by chance as in, and they sort of shrug and say it's just happened. Meaning it's not some known external cause. Errors when cells divide, however, they are expected. There are then a whole load of repair mechanisms, and so they should normally get mopped, either fixed, or if they can't be fixed, the cell should be forced to sort of kill itself, and these are the things that happen. Okay. This is just a sort of principle. It is useful to say. So cell division is a normal thing, okay? So one cell becoming 2 and then 2 becoming 4. That is a normal thing. And when cells divide, it's tightly regulated. They call it this thing the cell cycle, and there's a whole load of checkpoints that stop it progressing through the cell cycle and other sort of signals that promote cells to go through the cell cycle. But this is what cells do when they divide. They get the DNA, they copy the DNA, they sort of split the cell in 2, and you get this supposedly perfect copy of the DNA in both cells. So that's the normal process of cell division. Now on the next slide, it's sort of it's suggesting what happens when you get cancer. So you get a cell at the top that divides, but when it divides, I think the red on this is to illustrate an abnormal bit of sequence. And so that abnormal bit of sequence that we think is, it may be a driver mutation or it may be an unknown significance mutation, but it's now a cancer-specific mutation because it's acquired or it's passed down into the sort of daughter cells of the cancer. And if it is a driver mutation, so therefore it's kind of required to push the cells into this cancer behavior, all the daughter cells, i.e. all the cells that the cancer is made up of, will then have these mutations. We talk about things like tumor evolution, which is when you get one initiating mutation, and the assumption will be all the daughter cells will have that one mutation, and then you can get sort of secondary mutations that drive a different behavior of the disease and things like this. And so you can find different what we call clones of cells within a single person. And this is relevant when you're doing biopsies. You biopsy a primary tumour, a secondary tumour, multiple biopsies, or even multiple biopsies of one tumour, which people have done for kind of research purposes. And you can see how the genomics of the tumour changes and evolves over time. And conceptually, it's a kind of Darwinian evolution. You get these selection pressures. So if the mutation gives it a survival advantage, then it starts to populate the tumour. If the mutation is not really much good, then it gets kind of out-competed by other populations. But just go back one thing. So just one thing to mention is, if you're doing whole genome sequencing, you can see we've got your genome and cancer genome. So we look at the two. You sequence the patient's own genome, and we sometimes use the word germ line, like the sort of, they initiated the sperm cell germ line. And we sequence the cancer. And you sequence the two and then look for the difference. And that's how you kind of call your variants. And it's a variant from the germline. So that's kind of what you're looking for. So you sequence everyone twice when you're doing this whole genome sequencing. Okay. Okay, so yeah, cancer is lots of things. it's different things for different people. If it depends what kind of your area of biology is, you know, so if you're a cell biologist, everyone's got their own area that they think is their sort of area of study. There's probably 10 or 20 different characteristics that are required really for a normal cell to become a cancer cell. But uncontrolled cell division is one of them, because if you don't have uncontrolled cell division, you've not really got a cancer, you've just got a cell, which is tiny, and it's not going to do any harm to anyone. So clearly, uncontrolled cell division is one of the hallmarks of cancer, we'll call it. Okay. And I think maybe I got ahead of myself talking about his cars before, but just to reiterate, so you can get, if you've got if you've got a change in your DNA that changes the protein, and the protein promotes cell division in this instance, then you're promoting one of the traits of cancer. And if it's the opposite, so if the protein normally restricts cell division in this kind of thought experiment, then you mutate the protein and it stops working, then cell division is kind of unleashed. And so that's what, so it's probably not that important if it's too weird to get your head around, but when we look at what the effect of the mutation is on the protein, these are the kind of questions we ask. Does it affect the function? Does it promote the function? Does it suppress the function? And then we use what we know about the protein's normal function to infer whether that is then cancer promoting or not. And so that's I shan't reiterate it, but just to say Notch is a really interesting gene and protein, because so in adenoid cystic, we kind of talk a bit about Notch, don't we? Around 10% of 10 or 15% of patients might have a Notch mutation in the recurrent setting, and Notch actually can promote cancer if you get a gain of function or a loss of function. So it's got to be kind of, like the porridge in the Goldilocks story has to be just right, kind of not too hot, not too cold. Notch is like Goldilocks porridge, if you like that analogy. So it's got to be just right. So for instance, when we had our study of the Notch inhibitors for adenoid cystic with Notch gain of function, one of the side effects of the drug was skin cancers, squamous carcinomas of the skin, because Notch suppression is bad in other contexts. Okay, so it's important for lots of reasons, isn't it? But we talk about using tests as help to aid diagnosis. So we call them diagnostic biomarkers, diagnostic tests, to aid understand the disease course and to predict response to treatment. And so in adenoid cystic, half the people in the room probably know this, so MIB gene fusion, we don't really talked about gene fusions actually, but MIB gene fusion is used to aid the diagnosis of adenoid cystic carcinoma. Notch gain of function mutation informs the disease course and, to a degree, predicts response to treatment. So we know that patients without Notch gain of function mutation, you wouldn't even offer them these Notch targeted therapies. So if we move on, I think there's some slides just kind of talk about it in other examples. So this is actually the best example, so using genomics to develop new treatments, it's kind of high risk. The whole thing about whole genome sequencing has been going on for 10 years. And there aren't dozens of examples of targeted therapies that you can that are really good based on your results. But there are some examples. And in the world of salivary cancer, this example is probably the best example across all cancers, and it's a salivary gland cancer. Okay, so there's this tumor type called mammary analog secretory carcinoma, which is now often just called secretory carcinoma, because mammary analog just means like the breast, and there is also a secretory carcinoma of the breast. Okay, so if we just call it secretory carcinoma, and secretory carcinoma of the salivary gland, almost all patients have this NTRK gene fusion. In the same way, almost all patients with adenoid cystic carcinoma have a MIB fusion. Almost all patients with a mucoepidermal carcinoma have a MAML, MAML2 fusion. Okay, so in this particular salivary gland cancer, almost all patients have an NTRK fusion. So it's useful as a diagnostic biomarker, although a pathologist will probably say, yeah, but I could just diagnose it anyway by how it looks. But if you've got this gene fusion, you know it's a secretory carcinoma in the salivary gland. And we've talked about MIB and FIB in adenoid cystic, but I think NTRK is on the next slide, isn't it, as well? Oh, you know, this is the exciting thing. This is why we're doing it, is to try and develop new treatments, isn't it? So then there's a class of drugs that target this NTRK gene fusion. So that fusion, it's not actually a mutation, the whole sort of bit of the genome gets broken away and sticks to another bit of the genome and forms this, what you call a fusion protein, and it's that protein that's kind of cancer promoting. And there are drugs that target NTRK overactivity. And when those drugs were developed, they were developed in any tumour type, any bit of the body, but as long as they had a NTRK and NTRK gene fusion. And most patients respond to the drug. Response rates are kind of 80 to 90%. So response means tumour shrinkage. And most patients respond for years to the drug. Although it's not kind of 10 years, but it's like, over a year, over 2 years, some patients beyond. There are second generation, third generation NTRK inhibitors as well. So it's a really good example of a drug being developed that targets a genetic finding, and it doesn't matter what your tumour type is. And actually, salivary cancer is the sort of poster child, if that's the word, poster tumour, of this gene fusion. And so this is just a picture to try and explain this concept about what Sam's called classical trials, i.e. how we used to run trials, and this concept of a tumour agnostic trial, meaning agnostic meaning it's, in this case, irrespective of the bit of the body. So your classical trial in this terminology, you've got a picture of a stomach there, because you're not familiar with what they are. I think they're stomachs anyway. And what he's shown is you've got patients with stomach cancer, you've got a blue drug, and only the blue patient benefits. We don't even necessarily have a predictor, but you give everyone with stomach cancer this drug, and for whatever reason, it is a match for these, say, third of patients in that example. Or you've got a red drug, you give it to your lung cancer patients, and 1 in 3 of your lung cancer patients benefits, because for whatever unknown reason, often a third of the patients in that example are sensitive to the drug. And if you've got a tumour agnostic study, it doesn't matter what the tumour type is. You've got a stomach, a lung, a brain, whatever, but you've got a genetic finding like your NTRK gene fusion. And then you put all your patients on the trial and give them all the drug, irrespective of where it is in the body. And then you ask the question after the event. You say, well, does it benefit all tumour types equally or not? And it can actually be quite difficult to know, because if you look at the NTRK data for salivary gland cancer, so although I told you it's sort of the poster tumour, it's not that common for patients with secretory carcinoma to develop recurrent or metastatic disease. As many people probably know, you've probably never met anybody with secretory carcinoma, and I've met very few people with secretory carcinoma. And so there are very few people in the study. There was probably about 5 or 6 or something. So you then end up with really small numbers of patients with your particular tumour type, with your gene alteration, on the drug. But nevertheless, these drugs have been licensed. We've talked about this before in this group about the kind of drug development process. They've been tested, they've been licensed for any tumour type with the gene mutation, and they are reimbursed through our healthcare system. So that's like the hardest test. Not everything gets reimbursed. Lots of drugs are licensed but not reimbursed, but they are tested, licensed, reimbursed.